It is my pleasure to introduce Dr. Gschell, who is currently the head of Cryo-EM at BIMOVIS. He studied chemistry, biochemistry, and biophysics at the Albert Ludwigs University in Freiburg. For his doctoral work, he performed a full mechanistic analysis on a membrane-spanning ammonium sensor, and he has a deep technical expertise in microbiology, membrane protein purification and stabilization, as well as enzymatic and electrochemical analysis methods and cryo-EM. At BIMOVIS, he now specializes in end-to-end structure determination from sample optimization all the way through to data set processing strategies. And now I welcome Dr. Gschell to the screen and to take us away.
Thank you so much for the nice introduction. And hello everyone, and welcome to my talk about mass photometry in Cryo-EM sample preparation. So I work in a company that specializes on structural biology primarily. So this means that we combine experimental structure determination using all methods from X-ray crystallography to Cryo-EM and Cryo-ET with AI predictions in cases where structure determination is impossible, as well as high-end visualizations, for example, for stakeholder discussions or marketing. Finally, we do training and consulting on your structural biology projects, as well as helping you getting independent from services like ours. We are a small company that has formed about 3 years ago and is now in full production for about 2 years. And since then, we've worked on a variety of interesting different topics. I want to quickly highlight our team, specifically the 3 people interesting for structure determination projects, which are Dr. Florian Schneider, our head of protein crystallography, as well as Dr. Evgenia Zagori for our Cryo-ET projects, and of course myself. Our standard structure determination workflow evolves around purified protein. Usually clients come to us already with the protein at hand, either produced or, if you're very lucky, bought on commercially available markets. However, we do also specialize in producing more complicated samples ourselves, which is especially relevant for fragile membrane protein complexes and other transient samples that you want to analyze. I want to specifically highlight steps 2 and 3 of our workflow, since those parts are the ones that I'm specifically going to talk about in this presentation. Usually, whatever protein comes in, whether produced by us or sent in by a client, is first analyzed towards its quality to make sure that the protein we're dealing with is good enough for the later and sometimes quite expensive steps that follow afterwards. This is also especially important for complexation experiments that are done in-house, where you then, of course, have to further purify and again verify the quality of the resulting sample. And then the sample is ready for workflows like X-ray crystallography or Cryo-EM, both of which highly benefit from a pure and homogeneous sample, which is especially true in X-ray crystallography, where a polydispersed sample typically doesn't crystallize at all and stops you in your workflows. Looking a bit closer at these 2 steps, we do have the initial quality control, followed in many cases by a complexation, being with a different protein or with various small molecule ligands. We then go into size exclusion polishing, which is done both for samples that are not sufficient yet in terms of purity, or of course, if we just worked on them in the lab. And then we go into another round of mass photometry analysis before the sample is ready to go into the structure determination processes. I'm now going to talk about 3 different example cases where mass photometry was especially useful in the past to guarantee that the later and much more complex workflows have a good sample to start out with. So mass photometry is a very small, very short experiment where we use a minuscule amount of sample, and within just a minute or two of analysis can directly say whether the sample is good to go or not. This is especially relevant in time-sensitive workflows where we usually cannot afford a analytical size exclusion or a similar method, just to make sure that we can approach with the next steps. In our first case, we were working on a heterodimer complex of about 200 kilodalton. This was a request to solve the structure of two artificial binders. And as I told you before, our first step is always to analyze the sample as it comes into the lab. In this case, thankfully, we had very pure, very homogeneous protein to work with. One of about 110 kilodaltons and one of about 80 kilodaltons, then forming the expected 200 kilodalton heterodimer in a 1-to-1 ratio. Since this was already common knowledge, we then moved on and added the respective molar ratio together, running them over a size exclusion column to purify away the complex species from usually leftover monomer, since those concentration experiments, while being relatively accurate, typically never hit the point quite perfectly. So this is also what we see here. Basically, in those complex experiments, if nothing goes wrong, of course, you do end up with two peaks, one being the heavier complex peak coming more to the left, and one being leftover sample, which follows afterwards to the right. Looking at the expected species, we can therefore assume that we have a complex and a monomer peak, and the only thing that in this experiment caused a bit of worry was that the amount of monomer left over wouldn't really match the amount of protein we put into, since if we were at least halfway correct in the amount of protein used, we should not end up with such a significant monomer peak left over. Looking at the peaks in more detail, however, we see that both of these look very homogeneous and quite similar, with the only difference being a bit of a tailing end on the right for the second peak. But from a general point of view, both of these peaks look like samples that should behave quite well in a further structure determination experiment. However, to have this in a bit more resolution, we do always run mass photometry on those purified samples. And for the first peak, we do get what we expected, which is a above 200 kilodalton peak that is symmetric and relatively pure, with a small amount of artificial heterotetramer, which for this species of protein was to be expected, but the amount is neglectable. For the second peak, however, we see a much worse picture. While the overall Gaussian distribution fitting of the software here definitely just tried to estimate what it's finding here, we can clearly see that we do have a size distribution spanning all the way from a monomeric, roughly 100 kilodalton species to more than 230 kilodalton being likely a homodimer of the heavier of the two species. This is a huge problem in structure determination, since what we're looking at here is dynamic oligomerization. Basically, a portion of the protein that, while often enough in complex with either itself or its binding partner, runs at a relatively smooth peak on the column. Overall, you always, so at any point of time, have a mixed species of proteins in there that in size and in shape look surprisingly similar. If you have something like that on a Cryo-EM grid, you will have a very hard time distinguishing any single species out of it and solve any structure. So knowing that, we could optimize our workflows towards a cold and very fast transfer from purified sample to grid, and we were actually able to solve the structure even though we had this underlying difficulty. The second example is more of our usual business. Here we have a antigen antibody complex, or to be more specific, a antigen VHH complex. Basically, a VHH domain is a single binding domain taken from a antibody. So one of the four binding domains you would get in the complete protein, and it's pretty much the smallest, most stable part of an antibody that you can still utilize to show the full binding capacity of your developed species. So those VHH domains are commonly used in crystallization experiments and other situations where a lot of protein is needed and you don't want to work with the added complexity of the very flexible antibodies. In our case, this of course means that we have a species that is so small that we can neither really see it on a size exclusion column nor analyze it with the mass photometer. So for the initial experiment, we only could look at the antigen. This one is a 190 kilodalton monomer with, again, a very small amount of artificial dimer in the mix. But it looks fairly homogeneous and ready to go into complexation. Adding a excessive amount of the VHH domain to guarantee a large quantity of complex, we came up with, again, the typical two peaks of the supposed complex species and an excess of the smaller binding partner. And in purification, we leaned heavily towards the heavier left side of the main complex peak to make sure that we grab as much complex from that species as possible. Looking at the post-purification mass photometry analysis, we do see that under this high dilution in the mass photometry measurement, we do have some dissociation, which, thanks to the small size of the particle or of the VHH domain, is wrongly classified as a species of about 53 kilodaltons, followed by the about 200 kilodalton supposed complex, as well as a now even heavier dimer species at 450 instead of 400 kilodaltons in the mix. If we overlaid the main peaks, we can now see that some shift has occurred, specifically from about 193 kilodaltons to 204, which very closely aligns with the added mass of a single VHH domain. Thankfully, due to the low amount of sample required, I was also able to repeat that experiment and reliably get the same or a very similar shift in weight, giving me a very nice guarantee that what we're putting onto the grids is actually the complex species. So for the first example, I've put the last minute verification in. This is basically just another measurement, but it is in a way convenient that I could not reproduce with any other method to date. Basically, here we're working with a homodimer of 520 kilodaltons that was purified in-house. Again, we do see some sort of dissociation due to the dilution effect seen in standard mass photometry experiments, which means that we likely have at least some free monomer at 260 kilodaltons combined with another portion of the dimer at 522 kilodaltons in that mix, as well as some small impurities left over from purification. If we use this sample in Cryo-EM, the pilot data immediately was reason for caution. Basically, we expected to have primarily monomer and dimer in here, and especially the dimer with half a megadalton of size. should give a very large and easily identifiable particle in this data, even if pilot data is slightly lower resolution. However, we could only identify several monomer species particles, as well as a significant portion of protein that, while nicely averaging to the classes, so they seem to be rather homogeneous, are far too small for the species that we actually expected in the data set. This, of course, then leads up to the question of whether or not the protein is fragmenting within that final sample mix. What we used here then to solve that is that we could relatively easily analyze the mix not only after purification, where we have time and the protein is now finally ready for storage conditions, but also after adding, in this case, 3 different small molecules and quite larger ligands to it, and detergent that is used for orientation bias issues in sample prep. So we could create the grids, and then with the last leftover microliters, just put them back into the mass photometer, and within a minute between the last grid being frozen and this sample being analyzed, determine whether or not that final mix has altered the sample to the degree that we see in the data set. And looking at those results, we did see a significant size increase due to the various ligands binding to the complex. However, we do not see any visible fragmentation until the vitrification happened, which normally freezes everything that happens into place. And we can therefore conclude that the most likely outcome of our data set issues is a sorting on the glow discharge grid, which happens in several cases when different protein surfaces are exposed and therefore act differently to the highly hydrophilic metal surface of our sample holders. This is extremely convenient to know, since the reaction to fragmentation is vastly different to changes in glow discharge effects. So within just a very easy and quick to do experiment, we had an answer that would immediately lead us to a much better sample instead of testing various different parameters. So to sum up our main benefits from mass photometry, we do get a faster, cheap quality control, which means that within just a very short time and without wasting the sometimes very limited amount of sample, we can determine whether what we have at hand is good to go or needs further polishing. We do also, which is not shown in examples, but we tend to make use of it quite repeatedly, test whether ideal ratios and stoichiometry are correct for complexation experiments whenever the client is unsure in those regards. We can then test what came out of the size exclusion, which with much higher precision than the preparative scale SEC itself. And finally, as I just talked about, we can verify that what went onto the grid and was vitrified is exactly what we planned to have there. So we can clearly identify where several issues are coming from when we're looking at data sets quite a bit later. With that, I'm at the end of my talk, and I'm now going to hand over to Selma, who is going to talk more about mass photometry.
Thank you so much, Matthias. That was a wonderful talk. And hello again, everyone. I will be sharing my screen. Fantastic. I think everyone should be able to see my screen now. So hello again. I'm Selma. I'm a market development manager over at Refeyn. I've been with Refeyn for about 3.5 years now, and my favorite part of my job is working with our customers to understand where their analytical pain points are, and to see if how we can work together to see if we can accelerate and simplify those workflows with mass photometry. In this presentation, I'll be introducing Refeyn's newest mass photometer, MyMass MP, that has been designed with the user in mind to make sample checking even easier and faster than before. As Matthias so nicely showed us just now, mass photometry is an essential tool in the structural biology toolbox because it delivers novel insights into quality binding stoichiometry, assembly mechanisms, and sample quality and reagent quality for a range of biomolecules like proteins and nucleic acids. In comparison to traditional screening technologies, mass photometry provides single particle measurements that are extremely fast while using virtually no sample. This is why mass photometry is already widely used by scientists in both academia and pharma, resulting in over 1,500 peer-reviewed publications and mass photometers being used in 18 of the top 20 biopharma companies globally. Many structural biologists use the 2MP to check samples prior to expensive and time-consuming techniques like Cryo-EM. But when we speak to our users, we find that 2MPs are typically located in core facilities and not where samples are actually being produced and optimized. So for some of our users, that means if they're lucky, their 2MP is in a facility that is just down the corridor. But for others, that facility might be a few blocks away or even in a different city. And what we've heard from these users is that although they really like using mass photometry, they would really like to be able to use it in their own labs so they could use it every day instead of having to go to a different space in order to have access to mass photometry. And for this reason, we created MyMass MP. MyMass MP is our newest mass photometer. It is designed to allow users to quickly and easily check sample mass with enough resolving power to differentiate different oligomeric states. It's a mass photometer, so you still get all of the benefits of mass photometry, like direct single particle mass measurements in native conditions. And it has been designed to be used in every lab. And this is in terms of ease of use, a smaller footprint, and affordability. MyMass MP is an instrument that we designed from the ground up with the user experience in mind. So checking samples can easily fit into your everyday workflows instead of you having to work your sample checking around and making time for that in separate ways. So one feature that achieves this is the fact that you have no warm-up time with this instrument. So you can walk up, turn on the instrument, and take a measurement within a minute. And also to reduce the footprint, the system does not require any peripherals like a PC or a monitor. And the system comes with this touch screen. So you do all of your data acquisition and analysis in this one compact unit. And this allows you to make decisions on your samples quickly. And one of the things that onboard touchscreen allows us to do is it makes it easier to provide really clear instructions for our users. So you can remove quite a bit of training time that you would need for occasional users or newer users to the system. So with this touchscreen, we have this guided workflow with pictures to show you the setup of how to set up your consumables and then set up your measurement. And there's also extensive help and advice pages built into the software as well. And after the 1-minute measurement is complete, you have your data, and the system will give you a plot of the mass distribution of all of the molecules in your sample, as well as abundances of the different species in that sample in a table format. And this can be exported in standard PNG and CSV formats. And Refeyn ensures you have the complete solution. So we provide dedicated sample carriers and calibrate consumables that are available for you. So you have everything you need to do a measurement. In this video, you can see what a MyMass measurement looks like. Here, 30 microliters of a 10 nanomolar sample is being loaded onto the carrier slide. Pipetting is probably the most difficult step, so you don't need a subject matter expert in order to be able to check your samples every day. The measurement acquisition time is 1 minute. And over here, you can see the molecules that are being measured live. So you see each of those contrast signals is a molecule that's being measured, and then building that mass distribution plot out of thousands of molecule mass measurement events. And once the measurement is complete, you're going to get, as we are familiar, the mass distribution plot we discussed before, with the relative abundance of the different populations in there. Each measurement with MyMass typically uses nanograms of protein and will cost less than $6. MyMass MP can be used to rapidly check product purity and quality while conserving sample for downstream analytics. So in this example, say you have two products and you want to be sure which reagent is better to use for your downstream analytics. You can compare the purity and quality using MyMass in a minute and using virtually no samples. So you're conserving your sample for later analytics. And in this situation, we looked at two product offerings, one with a low catalytic activity and one with a high catalytic activity. And MyMass showed that the low activity product actually had impurities in there and a lower abundance of the functional tetramer species. So it's only 34% of the functional species in the low activity product versus 55% in the high activity product. And MyMass was able to give you this readout on purity and quality in a minute using virtually no sample. Another way you could use MyMass is for buffer optimization screening. So if you wanted to look at different conditions with your buffers, like salt concentration, or in this situation, looking at the effects of pH on stabilization of your oligomeric species. So here we used MyMass to screen the buffer pH conditions that stabilize dimeric thyroglobulin. And as you can see here, in the conditions more basic than 4.6, the thyroglobulin was predominantly dimeric. And MyMass was able to enables rapid screening, buffer screening, with measurements that take minutes using virtually no sample. So MyMass MP is the newest mass spectrometer available to support structural biology studies that enables rapid protein sample checking and easy protein sample checking. But this system fits into an existing portfolio of products that we offer for structural biologists. And if you want to do more advanced analytics, so if you want more information than just purely sample checking, you want detailed information on binding stoichiometry or complex assembly, then you would probably prefer using the 2MP product line, as the 2MP product line enables collecting more comprehensive data sets on a wider range of sample types. You can also work with nucleic acids, and you're not limited to protein work there. And then also with the 2MP, you are able to get add-on features such as an automation systems. So with the auto, you can enable walkaway time for 24 sample measurements, or you can get a rapid dilution system with Mass Fluidics HC. You can extend the concentration range of measurements up to 50 micromolar concentrations. So if you want clarity, speed, ease of use, and confidence in your analytics, we urge you to consider mass spectrometry to bring all of the advantages we've discussed today to your workflows.